Imagine standing on a planet that no human has ever touched. The sky above you is not blue. It's a dusty, bruised shade of red, like a sunset frozen in time, stretched across the whole horizon. The ground beneath your boots is cold, rust-colored and cracked. There's no bird song, no wind you can breathe. The real question isn't how we get to Mars. It is what happens to a human being once they do. If you were the first human to reach Mars, what is the very first thing you would do? Let us know in the comments. I will pin the most creative answer. Now let's begin. Here's a thought that might stop you cold. The first human on Mars won't see that pale blue dot the way Voyager did. They'll see something different. From the surface of Mars, Earth will look like a bright star, a blue-white point of light hanging in a pinkish Martian sky. On a clear night, you could cover it with your thumb. Everything you've ever known, everyone who has ever loved you, every mountain and ocean and memory reduced to something you can blot out with your fingernail. And that moment, that precise psychological instant, is where this story really begins. Let's talk about distance in a way that actually means something to your brain, because our minds are not built to process astronomical numbers the way they process, say, a walk to the corner store. Mars is not our neighbor in any friendly sense of the word. Depending on where the two planets are in their orbits, the distance between Earth and Mars ranges from about 38 million miles at the closest, what astronomers call opposition, to as far as 250 million miles when they're on opposite sides of the Sun. On average, you're looking at roughly 140 million miles. Light, the fastest thing in the universe, travels at about 186,000 miles per second, and yet, a beam of light takes between 3 minutes and 22 minutes just to cross the gap between Earth and Mars, depending on orbital positions. A radio signal, the kind used to communicate with spacecraft, travels at the same speed. What that means in practice is almost impossible to fully appreciate until you sit with it. If the first human on Mars calls home and says, "I made it, I'm okay," the people on Earth won't hear those words for somewhere between 3 and 22 minutes. And the reply? That takes just as long to travel back. A two-way conversation, the kind you have with a friend in seconds, could take 44 minutes round trip. At maximum distance, nearly 45. NASA's own Human Research Roadmap identifies this communication delay as one of the most serious psychological challenges of a Mars mission. Not because people can't wait, but because in a crisis, a medical emergency, a mental breakdown, a life or death decision, those minutes stretch into something terrifying. The crew on Mars will be, in the most literal sense, on their own. Now think about what that does to a human mind. We are, at our deepest biological core, social creatures. Our nervous systems evolved over hundreds of thousands of years in tight-knit groups, tribes, families, communities. We are wired to reach out, wired to connect. Our brains release oxytocin, the bonding hormone, through touch, through eye contact, through hearing a familiar voice. Loneliness, real clinical loneliness, doesn't just feel bad. Research shows it raises cortisol levels, disrupts sleep, weakens the immune system, and over time can cause measurable changes in brain structure. And no crew member heading to Mars will escape some version of it. But before we even talk about what happens on Mars, let's talk about how you get there, because the journey itself is its own extraordinary transformation. Using current propulsion technology, a one-way trip to Mars takes somewhere between 6 and 9 months, depending on the launch window. Missions are typically planned around what's called a Hohmann transfer orbit, a fuel-efficient trajectory that takes advantage of the gravitational relationship between Earth and Mars. These optimal launch windows occur roughly every 26 months, when the two planets are aligned in a way that minimizes travel time and fuel, so a full round-trip mission, launch, travel, surface stay, and return, could last anywhere from about 2 years to nearly 3. That's 34 months at a minimum for a conjunction-class mission, which involves spending around 500 days on the surface while waiting for the next launch window back to Earth. 500 days on a planet where the temperature drops to -80 degrees Celsius at night, where the atmospheric pressure at the surface is less than 1% of what you breathe on Earth right now, where a dust storm can darken the sky for weeks, where cosmic radiation pelts the surface because Mars has no global magnetic field and only a thin sliver of an atmosphere to shield against it. Now, here's where it gets fascinating. Because during those 6 to 9 months of travel through deep space, before you even land, something remarkable and alarming is already happening to your body. In 2012, NASA's Curiosity rover was launched toward Mars, carrying an instrument called the Radiation Assessment Detector, or RAD. Its job wasn't to study Mars, not yet anyway. Its job was to measure the radiation environment during the cruise phase of the trip. The data it sent back was sobering. A round-trip flight to Mars exposed the hypothetical crew to a radiation dose of approximately 0.66 sieverts. NASA's career radiation limit for astronauts is 1 sievert total. Just the trip there and back would nearly max out that limit, and that's before counting any time spent on the surface. To put that in perspective, the average person on Earth receives about 0.003 sieverts of radiation per year from background sources. A single round trip to Mars, under current shielding technology, would expose an astronaut to more than 200 times that annual dose. But radiation isn't the only threat in transit. In the microgravity of deep space, the human body begins to change in ways that would have been unimaginable to the astronauts of the early Apollo era. Bone density drops. Without gravity's constant pull, bones don't experience the mechanical stress they need to stay strong, and the body, ever efficient, begins to break them down. Studies of astronauts returning from long-duration missions aboard the International Space Station have shown bone loss rates of 1 to 2% per month in load-bearing bones, the hips, the spine, the legs. That's 10 to 20 times faster than the bone loss seen in elderly people with osteoporosis on Earth. Muscles shrink. Without the constant low-level effort of simply standing upright and moving through a gravitational field, muscle mass atrophies rapidly. The heart, itself a muscle, actually changes shape in microgravity, becoming more spherical. Fluid shifts toward the head, increasing pressure on the optic nerve and causing a condition called spaceflight-associated neuroocular syndrome, or SANS, which can impair vision. In June of 2024, researchers at University College London's Department of Renal Medicine published a landmark study warning that galactic radiation combined with microgravity poses serious risks to kidney function on a Mars mission. Their findings suggested that the longer an astronaut is exposed to these combined stressors, the more likely they are to develop kidney damage, potentially requiring dialysis before they even make it home. This is the body fighting a war it was never designed to fight. And yet, and this is where I want you to lean in close, because this is the part that changes everything, the human body is not passive in this story. It is not simply a machine being damaged by a hostile environment. It is an adaptive, dynamic, astonishingly creative biological system that has survived ice ages, famines, pandemics, and every other catastrophe this planet has thrown at it for roughly 300,000 years. The first human on Mars will not just be a victim of their environment. They will be, in the most profound scientific sense, the beginning of something new. Think about that for a moment. Every astronaut who has ever lived aboard the International Space Station has returned to Earth with data, with biological changes, with psychological transformations. Astronaut Scott Kelly spent 340 days aboard the station as part of NASA's famous twin study. the most comprehensive examination of long-term spaceflight effects ever conducted. His identical twin, Mark Kelly, stayed on Earth as a genetic control subject. When Scott returned, scientists found changes in his gene expression, the way his DNA was being read and translated, that differed significantly from Mark's. Some of those changes persisted for months after landing. Others may be permanent. His telomeres, the protective caps on the ends of his chromosomes, associated with aging and cellular health, actually lengthened during his time in space, something that surprised researchers deeply. They expected the opposite. What that tells us is this: space doesn't just break the body down, it also switches things on, it activates pathways, it forces adaptations, it asks questions of the genome that Earth's gravity has never thought to ask. The first human on Mars won't just be exploring a planet, they'll be exploring the outer edge of what it means to be human. That's where we're going in this series. Over the next 9 parts, we're going to travel through every dimension of this transformation. From the cellular to the cosmic, we'll look at what radiation does to DNA, and why some researchers believe low-dose radiation might actually trigger protective biological responses. We'll explore the psychology of isolation in ways that will make you rethink your own need for connection. We'll examine the overview effect, that well-documented phenomenon where astronauts undergo a profound shift in consciousness upon seeing Earth from space, and ask, what happens when that blue dot is so far away you can barely find it? We'll talk about the circadian rhythms that govern your entire biology, your sleep, your hormones, your immune function, and what happens when those rhythms lose their anchor, because Mars days are 24 hours and 37 minutes long, not 24. 37 extra minutes every single day, compounding over 500 days into a biological drift that science is only beginning to understand. We'll look at the microbiome, the trillions of bacteria that live inside you right now, influencing everything from your mood to your immune system, and how that ecosystem might change in an environment with different gravity, different radiation, different dust. We'll ask whether the first human on Mars will feel something no human has ever felt before, not just the overview effect, looking back at Earth, but something entirely different, something we don't even have a name for yet. What happens to the human psyche when you look up at a night sky from another world and see the Milky Way from a new angle? When you realize, for the first time in the history of our species, that home is not where you are standing. And finally, we'll ask the biggest question of all. Is this the beginning of our evolution into a multiplanetary species? And if it is, what does that mean for what we are, for what it means to be human? Because here's the thing about us. We are not rocks. We are not fixed. We are the universe becoming aware of itself, borrowing atoms from ancient stars, arranging them into nervous systems curious enough to ask where they came from. Every element heavier than hydrogen in your body was forged inside the heart of a dying star. The iron in your blood was hammered into existence in a supernova explosion billions of years before Earth existed. You are quite literally made of the cosmos, and the cosmos, in a very real sense, is made of you. When the first human steps onto Mars, they won't be leaving the universe behind. They'll be carrying it with them, in every cell, every breath, every heartbeat, across the void between two worlds. That's not poetry. That's astrophysics. That's biology. That's the most extraordinary story our species has ever been part of. And it's just getting started. Let's start with something small, impossibly small, smaller than anything you can see, smaller than anything you can feel, but powerful enough to change the entire course of a human life. Let's start with a single strand of DNA. Right now, inside every cell of your body, there are roughly 3 billion base pairs of genetic code, the complete instruction manual for everything you are. Your eye color, your immune system, the way your neurons fire when you hear music you love, the proteins that repair your muscles after exercise. All of it encoded in a molecule so thin that if you stretched out the DNA from a single human cell end to end, it would measure about 6 feet long. And yet it's coiled so tightly it fits inside a nucleus far smaller than the period at the end of this sentence. That molecule is extraordinary. It is also fragile in ways that matter enormously when you're talking about a journey to Mars. Here on Earth, you are protected from the worst of space's radiation by 2 invisible shields. The first is Earth's magnetic field. The magnetosphere, a vast bubble of electromagnetic force generated by our planet's spinning iron core, extending tens of thousands of miles into space. It deflects the most energetic charged particles the Sun and the galaxy throw at us. The second is our atmosphere, roughly 60 miles of air pressing down on everything, absorbing and scattering radiation that gets through. Mars has neither of these in any meaningful form. Mars lost its global magnetic field billions of years ago, most likely when its smaller iron core cooled and stopped convecting, shutting down the dynamo that generates a magnetosphere. Without that field, the solar wind, a constant stream of charged particles blasting outward from the Sun at roughly 1,000,000 miles per hour, began to strip away the Martian atmosphere over hundreds of millions of years. Today, Mars has an atmosphere that is about 95% carbon dioxide and so thin that its surface pressure is less than 1% of Earth's at sea level. That's not enough to stop much of anything. On the surface of Mars, you would be bombarded by 2 main types of radiation. The first is solar energetic particles, bursts of high-energy protons and electrons thrown off by solar flares and coronal mass ejections. These events are somewhat predictable, and a well-shielded habitat could protect a crew during the worst of them. In September of 2017, NASA reported a massive solar storm that temporarily doubled radiation levels on the Martian surface, producing an aurora 25 times brighter than anything previously observed there. For an astronaut in a spacesuit during that event, the exposure would have been significant. The second type is far more insidious, galactic cosmic rays. These are high-energy particles, mostly protons, but also heavier atomic nuclei, that have been accelerated to near light speed by supernova explosions and other violent events across the galaxy. They come from every direction, all the time, and no amount of warning helps because you can't predict them. They punch straight through thin walls. They punch through you. When a galactic cosmic ray, particularly a heavy ion like iron or silicon, passes through your body, it doesn't just deposit energy the way an X-ray does. It tears through the molecular architecture of cells like a bullet through paper, leaving a trail of broken chemical bonds and shattered DNA. Scientists call this track damage, and it is fundamentally different from the kinds of radiation we deal with here on Earth. In medical imaging or even nuclear plant environments, most of your cells can repair this kind of damage. Your body has remarkably sophisticated DNA repair mechanisms honed over billions of years of evolution on a planet that, while shielded, still receives background radiation constantly. These molecular repair crews, enzymes and protein complexes with names like BRCA1 and 2, the ones you might have heard about in the context of cancer risk, scan your genome constantly, finding breaks and stitching them back together. But they're not perfect, and under the sustained high-intensity bombardment of deep space and the Martian surface, the error rate climbs. Some of those errors are harmless, some lead to cells that simply stop dividing, but some, the scary ones, lead to mutations, to cells that divide uncontrollably, to cancer. NASA's radiation limit of 1 sievert over an astronaut's career was set specifically because that exposure corresponds to roughly a 3% increase in lifetime cancer risk, a threshold the agency determined was acceptable given the benefits of the mission. A Mars trip gets dangerously close to that ceiling on the journey alone, before anyone sets foot on the surface. But here's where the science takes an unexpected turn, one that feels almost like the universe has a sense of irony. Not all radiation damage is straightforward destruction, and not all of the body's responses to it are purely defensive. There is a phenomenon that researchers have been studying for decades called radiation hormesis, the hypothesis that low doses of ionizing radiation might actually stimulate protective biological responses rather than simply causing harm. The idea is rooted in the same principle that makes vaccines work. A small challenge to the system can prime it to handle larger challenges more effectively. The evidence for radiation hormesis at the doses relevant to a Mars mission is genuinely contested. Some studies on cells and on animal models suggest that low chronic radiation exposure triggers upregulation of DNA repair pathways, essentially training the molecular repair machinery to work faster and more accurately. Others show clear cumulative damage with no obvious protective benefit at higher doses. What's not contested is this: astronauts who return from long-duration missions are not simply damaged versions of their pre-flight selves. They are changed, sometimes in ways that alarm physicians, sometimes in ways that surprise everyone. Scott Kelly's twin study, which NASA conducted between 2015 and 2016 and published its comprehensive results in 2019 in the journal Science, gave us the most detailed portrait yet of what space does to a human body over time. For 340 days, Scott lived and worked aboard the International Space Station while his twin brother Mark stayed on Earth as a genetic control. They ate similar diets, had similar sleep schedules, stayed in regular contact. The goal was to isolate the effects of spaceflight as cleanly as possible. The findings were, in the words of one researcher, striking. Scott showed changes in the expression of over 7% of his genes. His gut microbiome, the community of bacteria living in his digestive tract, shifted dramatically, with different species becoming dominant. His cognitive performance, measured by reaction time and accuracy tests, declined slightly during flight and took months to fully recover after landing. His telomeres, the protective end caps of his chromosomes associated with aging, lengthened in space. A counterintuitive finding, since telomeres generally shorten as we age, and then rapidly shortened again upon his return to Earth. His carotid artery walls thickened, his vision changed, likely due to increased intracranial pressure caused by fluid redistribution in microgravity. His DNA itself showed patterns of methylation, chemical tags that switched genes on or off, that differed from marks in hundreds of locations. Some of these changes resolved after landing. Others persisted for the full 18 months of post-flight monitoring. A handful appear to be permanent. What does this tell us? It tells us that the human body, when placed in the environment of space, does not simply endure. It actively responds, it negotiates, it recalibrates. It turns dials and flips switches in the genome that evolution never intended to be touched, because evolution never anticipated this situation. And for the first human on Mars, on a mission not of 340 days but potentially of 900 or more, those negotiations will go much, much further. Now let's talk about the brain, because this is where the story gets truly intimate. Your brain is, without question, the most complex object in the known universe. 100 billion neurons, 100 trillion synaptic connections, an organ that consumes roughly 20% of your body's total energy while representing only about 2% of your body weight. It is the seat of your consciousness, your memories, your sense of humor, your fear of the dark, your capacity to love another person, and it is exquisitely sensitive to the environment it lives in. One of the most concerning findings in recent space medicine research involves what cosmic radiation does to the central nervous system. In studies conducted by researchers at the University of California, Irvine, using simulated galactic cosmic ray environments at NASA's Space Radiation Laboratory at Brookhaven National Laboratory in New York. Mice exposed to the kinds of radiation they'd encounter on a Mars trip showed significant cognitive impairment. Memory formation was disrupted. Spatial navigation, the ability to find your way through an environment, declined. The animals showed signs of what the researchers described as cosmic ray dementia, not a clinical term, but an evocative one. The specific culprit in these animal studies was iron-ion radiation, one of the most biologically damaging components of galactic cosmic rays because of its high mass and high charge. Iron ions cause particularly severe tract damage through neural tissue, and unlike the softer cells elsewhere in the body, neurons are largely non-dividing. They don't replace themselves easily. When they're damaged, the effects tend to be cumulative and lasting. Now, to be absolutely clear, these are animal studies, and mice are not humans. The radiation doses used were compressed into short experimental periods rather than spread over years the way a Mars mission would unfold. Extrapolating directly to human astronauts is something researchers are careful not to do carelessly, but the biological mechanisms are real, and the implications deserve serious thought. A mission to Mars doesn't just ask your brain to function normally under stress. It asks your brain to function at the absolute limit of human capacity, making complex engineering decisions, managing interpersonal dynamics in a tiny enclosed space, performing medical procedures, conducting scientific research, all while being slowly irradiated, sleep disrupted, nutritionally constrained, and emotionally strained in ways no human being has ever experienced before. The first Martian astronaut won't just need to be physically fit. They'll need a mind that can bend without breaking, adapt without losing coherence, and find meanings in circumstances that have no precedent in the entire history of human psychology. And here's something that rarely comes up in the mainstream conversation about Mars missions, something that sits at the fascinating intersection of biology and philosophy: the role of the human microbiome. Inside your gut right now live approximately 38 trillion microorganisms, bacteria, archaea, viruses, fungi, collectively weighing about the same as your brain. This community, built up over your lifetime from birth, is so intimately integrated into your biology that many scientists now refer to it as a second genome. These organisms help digest your food, produce vitamins, regulate your immune system, synthesize neurotransmitters including serotonin, with about 90% of your body's serotonin actually produced in the gut, and even influence your mood and behavior through what's called the gut-brain axis. What happens to that ecosystem when you leave Earth? We know from studies on International Space Station crews that the microbiome shifts in space. The diversity of bacterial species tends to decrease, a generally bad sign, as microbial diversity is associated with better health outcomes. Pathogens, bacteria that cause disease, can become more virulent in microgravity. Some studies have shown that common bacteria like Salmonella become up to 3 times more infectious when grown in simulated microgravity. At the same time, the human immune system itself changes in space. White blood cell populations shift. Dormant viruses, ones that have been living quietly in the bodies of astronauts for years, like the herpes simplex viruses, begin to reactivate and shed at higher rates. The body's surveillance system, normally vigilant, seems to lose some of its edge. On a Mars mission, the consequences of getting sick are immense. There are no hospitals, no emergency evacuations, no pharmacists. The crew will carry a medical kit, pharmaceutical supplies, and emergency surgery protocols, but for anything serious, they are profoundly alone. This is why a December 2025 report from the National Academies of Sciences, Engineering and Medicine listed determining the impact of the Martian environment on crew physiological, cognitive, and emotional health as one of its top priorities for the first human mission to Mars, not as an afterthought, not as a box to check, as a core scientific objective of the mission itself. Because the first human on Mars won't just be a geologist or an engineer, they'll be a living experiment, a data point in the most important longitudinal study of human biology ever conducted. And what they teach us about radiation, about immunity, about the microbiome, about the adaptability of the human genome under extreme conditions, could reshape medicine on Earth just as profoundly as it shapes the future of space exploration. Let's close this section with a thought that might reframe everything you've just heard, every single thing that sounds threatening about this journey. The radiation, the bone loss, the immune disruption, the cognitive stress is, at its deepest level, a conversation between the human body and a new environment, a negotiation, an argument even, your genome arguing with the cosmos about what it means to be alive. And here's the thing about arguments. Sometimes you come out of them changed in ways that make you stronger, wiser, more capable than you were before. The human body has been having this kind of argument with hostile environments for as long as we've existed. When our ancestors migrated into cold northern climates, their bodies adapted over generations, lighter skin to absorb more vitamin D in low sunlight environments, genetic variants that processed fat differently, altered immune responses to new pathogens. When populations moved to high altitude, like the Tibetan Plateau, genetic changes in hemoglobin expression allowed them to thrive in thin air that would incapacitate a lowland visitor. Evolution, when it has time, is incredibly good at this. The difference with Mars is that we don't have generations. We have one person, one body, one mission. And the adaptations, if they come, will have to come fast, but they may come. And if they do, they won't just be a footnote in a medical report. They'll be the first chapter of something entirely new. The first human on Mars may not return to Earth entirely human, in the old, narrow sense of that word. They may return as something our species has never been before. Not alien, not superhuman, but expanded, stretched, tested at the edges and found, perhaps, to contain more than we ever imagined. That is not a warning. That is a wonder. Picture this: 6 people. One habitat, 520 days. No windows to the outside world, no fresh air, no ability to leave, no real emergencies. But no real escape either. The clock ticks, the walls stay the same, the faces around the dinner table never change. This was Mars 500 between 2010 and 2011, 6 male volunteers, 3 Russians, a Frenchman, a Chinese national, and an Italian Colombian, climbed into a series of interlocking steel modules at the Institute of Biomedical Problems in Moscow and sealed the hatch behind them. The facility had no view of the sky, no connection to the outside beyond highly regulated, delayed communications. Their mission was to simulate, as faithfully as possible on Earth, the psychological experience of a crewed mission to Mars. 520 days. That's not a weekend retreat. That's not a research posting in Antarctica. That's longer than the American Civil War lasted. Longer than it took the Allies to go from D-Day to the fall of Berlin. 520 days in a metal box, with 5 other people going nowhere. And what the researchers found when they analyzed the data changed how scientists think about the human mind in isolation forever. On the surface, the crew did remarkably well. There was no dramatic breakdown, no mutiny, no one had to be sedated or removed. They completed their simulated mission protocols, conducted their research tasks, held it together. By most outward measures, Mars 500 was a success. But beneath that surface, the data told a more complicated, more human story. One crew member gradually shifted his sleep cycle. Over weeks he drifted, sleeping later and later, waking later and later, until he was operating on a roughly 25-hour day rather than a 24-hour one. He became increasingly sedentary. His activity levels dropped. His engagement with the mission gradually pulled back, not dramatically, not suddenly, but like a tide going out, slow, quiet, and hard to stop once it started. Others showed episodes of what researchers described as psychological withdrawal, periods of social disengagement, reduced communication, emotional flattening. These weren't episodes of obvious depression in the clinical sense. They were subtler, a person sitting a little longer in silence, responding to questions with fewer words than usual, choosing to spend more time in their private quarters. Sleep disturbances affected most of the crew at various points. And here's why that matters. Sleep isn't just rest. Sleep is when your brain consolidates memories, clears metabolic waste products from neural tissue, regulates emotional responses. A well-rested mind can handle the same stressor that a sleep-deprived mind crumbles under. And on a mission where every decision carries potential life or death consequences, cognitive impairment from sleep disruption is not a minor inconvenience. It's a mission-critical vulnerability. The Mars 500 study also revealed something called behavioral health monotony, a gradual erosion of motivation and engagement that comes not from any single dramatic event, but from the relentless sameness of an isolated environment. The habitat didn't change. The food rotated through the same cycle. The faces across the table were always the same faces, day after day after day. Psychologists studying the data noted that this kind of monotony affects the brain's reward system in ways that are, neurologically speaking, similar to depression. The dopamine pathways that drive motivation and pleasure begin to quiet down when they're no longer receiving novel stimulation. The brain, in a very real sense, gets bored at a chemical level. And a bored brain is a dangerous brain on a Mars mission. Now here's what makes the real Mars mission categorically different from anything Mars 500 could simulate, and why the psychological stakes are so much higher than the study could fully capture. In Moscow, the crew knew they could leave, not easily, not without consequences to the mission, but the hatch was there, the outside world was there. If something went truly catastrophic, help was minutes away. The psychological weight of a genuine point of no return was absent. On Mars, there is no hatch that leads anywhere survivable. There is no quick rescue. The closest any help could possibly come is at the next launch window, which might be 26 months away. The communication delay alone, that 3 to 22-minute gap between sending a message and receiving a reply, fundamentally transforms the crew's relationship with Earth in ways that are difficult to overstate. NASA's own research identifies this as one of the 5 main hazards of deep space travel: distance from Earth, not just the physical distance, but the psychological distance, the feeling of being cut off, of being unreachable, of having problems that cannot be solved by the most sophisticated support network humanity has ever assembled, simply because that network is too far away to respond in time. Astronaut Scott Kelly, who spent nearly a year on the International Space Station, spoke about the psychological adjustment required even at that relatively modest distance. He described a phase familiar to most long-duration space crew members, sometimes called the third quarter phenomenon. Research on isolated environments, from Antarctic research stations to submarine deployments to space stations, consistently shows that morale and motivation hit their lowest point not at the beginning of an isolation period, not at the end, but at roughly the three-quarter mark. 3/4 of the way through, the initial excitement has long since faded. The end is visible but still agonizingly far away. Boredom, frustration, and interpersonal friction peak. This is when the real test of character arrives. On a Mars mission, the third quarter of a 2.5-year round trip falls somewhere around the 18th or 19th month, deep in the surface stay, surrounded by rust-colored rocks, with a communication delay that makes real emotional support from Earth feel more like reading letters from a distant friend than having a genuine conversation. But let's go deeper than behavioral patterns and sleep cycles. Let's talk about something that has only recently come into sharp scientific focus: the relationship between physical isolation and the actual structure of the human brain. Neuroscientists at the Alfred Wegener Institute in Germany studied a group of researchers who spent 14 months at an Antarctic research station, one of the most Mars-analog environments on Earth in terms of isolation and confinement. The researchers went in for brain scans before deployment and again after returning. The results were published in the New England Journal of Medicine in 2019. The Antarctic crew showed a measurable reduction in gray matter volume in specific brain regions, including the dentate gyrus of the hippocampus, an area crucial for learning, memory, and spatial navigation, and the prefrontal cortex, which governs decision-making, impulse control, and social behavior. They also showed reduced levels of brain-derived neurotrophic factor, or BDNF, a protein sometimes called miracle grow for the brain. Because of its role in supporting the growth and survival of neurons, reduced BDNF has been associated with depression, cognitive decline, and reduced neuroplasticity, the brain's ability to adapt and rewire itself in response to experience. And here's the haunting part: the researchers in Antarctica had a sky. They had seasons, though extreme ones. They had the natural world outside, however brutal. They had a horizon. The crew on Mars will have a horizon too, a stark alien one. But the sky will be the wrong color. The sun will be noticeably smaller, receiving only about 43% as much solar energy at Mars as at Earth. The terrain will be alien in a way that doesn't soothe the nervous system the way a familiar landscape does. There will be no trees, no rain, no smell of soil after a storm, no birdsong. Your brain needs novelty, yes, but it also needs beauty. It needs the kind of sensory richness that Earth provides almost automatically. And Mars, for all its stark magnificence, is not going to provide it. Now hold that thought, because here is where the story pivots in a direction that genuinely excites me. Because isolation and confinement are not only sources of suffering. Under the right conditions, in the right minds, they can be sources of something else entirely, something profound. There is a long tradition in human culture of people who sought out isolation deliberately, not as punishment but as a path. Monks in mountain monasteries, desert fathers of early Christianity, indigenous vision quests, philosophers who withdrew from society to think more clearly. What they were after, in different ways, was a confrontation with themselves that ordinary life's noise makes almost impossible. Forced isolation strips away the distractions, the social performances, the endless stimulation, and what it leaves at the bottom is you, your actual thoughts, your actual values, your actual relationship with existence. Research on long-duration space crews and Antarctic expeditioners has documented genuine instances of this. Alongside the behavioral withdrawal and the sleep disruption, some crew members report profound psychological transformations. Deepened relationships with crewmates, forged by shared hardship in a way that normal social bonds rarely achieve. A clarification of values, a stripping away of what doesn't matter, leaving only what does. A shift in perspective so profound that returning to ordinary life feels, for a while, like visiting a foreign country. Astronaut Peggy Whitson, who holds the American record for cumulative time in space at approximately 665 days, has spoken about how her extended missions changed her relationship with time, with priority, with what she considered worth caring about. She didn't describe it as suffering, though there was suffering. She described it as refinement. Chris Hadfield, the Canadian astronaut and commander of the International Space Station, wrote in his book that the experience of space taught him something he couldn't have learned any other way. That fear, managed properly, doesn't diminish you. It clarifies you. These are not small observations. They point to a genuine psychological dimension of long-duration spaceflight that goes beyond the clinical checklists of risk factors and behavioral health metrics. They suggest that the isolation of a Mars mission, genuinely unprecedented in its depth, might be capable of producing states of mind that no human being has ever accessed before. Let's talk about something called the overview effect, because it connects directly to what the first Martian might experience, and it's one of the most documented, most consistent, and most philosophically significant findings in the entire psychology of spaceflight. The term was coined by author Frank White, who interviewed dozens of astronauts about a perceptual shift many of them described after seeing Earth from space. When astronauts look down at our planet, this thin atmosphere, ocean-wrapped marble suspended in the void, they frequently report a sudden, overwhelming realization of interconnectedness, of fragility, of the arbitrary nature of all the borders and conflicts and divisions that seem so important from the ground. Apollo 9 astronaut Rusty Schweickart described it as a feeling of unity with everything he could see. NASA astronaut Edgar Mitchell, who walked on the moon during Apollo 14 in 1971, described an explosion of awareness, an overwhelming sense of oneness and connectedness accompanied by an ecstasy that he compared to a transcendent religious experience, though he was not a religious man in any traditional sense. Mitchell was so profoundly affected that he later founded the Institute of Noetic Sciences, dedicated to studying the intersection of human consciousness and scientific inquiry, a life trajectory he directly attributed to that moment, looking back at Earth from the lunar surface. More recently, actor William Shatner, who flew briefly to the edge of space on a Blue Origin rocket in 2021, delivered a genuinely striking description of what he saw. He talked not about beauty or excitement, but about a sudden, visceral sense of grief, of seeing the thin blue line of Earth's atmosphere as impossibly fragile, heartbreakingly thin, a barely there membrane between everything that lives and the cold indifference of space. He was visibly shaken, and he wasn't performing. Scientists have studied the overview effect with increasing rigor in recent years. Research published in the journal Frontiers in Psychology in 2022 analyzed interview data from 24 astronauts and found consistent patterns: a sense of cognitive shift, an expansion of self-concept, an increased feeling of universal connectedness, and a deepened commitment to environmental stewardship and human unity. These weren't just nice feelings, they were enduring, life-altering psychological reorganizations. Now, here is the question that no one has yet been able to answer, because no human being has ever been in a position to find out. What happens to the overview effect when Earth is no longer a magnificent sphere filling your window? From the surface of Mars, Earth is not a beautiful blue marble. It's a dot, a bright, pale dot in a pinkish-brown sky. A point of light you could easily mistake for a star if you didn't know where to look. Does the overview effect still happen when home has shrunk to the size of a period at the end of a sentence? Does it become more powerful, the distance amplifying the sense of fragility and preciousness? Or does it transform into something different, something we don't have a name for yet? A feeling of being adrift, of being cut loose from the gravitational pull, not just of Earth's mass, but of Earth's meaning? Or does it become something entirely new? A Mars effect, if you will, where standing on a second world for the first time, looking up at a sky you've never seen before, triggers a cognitive shift, not of connection to what you left behind, but of expansion into what you've become. We genuinely don't know, and that unknowing is one of the most thrilling scientific questions of our time. There is one more psychological dimension of a Mars mission that rarely gets discussed in public, and I want to bring it up because it matters. Both scientifically and humanly, the question of grief, the first crew to Mars will, in all likelihood, experience loss during their mission. Not necessarily death, though that possibility cannot be excluded, but the loss of ordinary human experiences that the rest of us take entirely for granted. The birth of a child they can't hold, the death of a parent they can't say goodbye to in person, a friend's wedding happening 200 million miles away that they watch in a video file with a communication delay long after the cake has been eaten and the guests have gone home. These moments of disconnection from the ongoing story of the people they love will accumulate. They will compound. And there is no protocol in any mission plan, not NASA's, not SpaceX's, not anyone's, that fully addresses the psychological weight of that kind of grief. What we do know from research on Antarctic expedition members and long-duration station crew is that the quality of a person's relationships, particularly their most intimate ones, is among the strongest predictors of how well they cope with isolation. Not their fitness, not their IQ, not their technical training, the depth and health of their personal bonds, which means that the first human on Mars will carry their most important relationships with them, not in their luggage, but in their nervous system, in the letters they reread, in the photographs pinned to the wall of their habitat, in the voice messages they listen to again and again during the long Martian nights. And this, in a strange and beautiful way, circles back to what we are talking about at the broadest level. The first Martian won't be a machine, they won't be a superhero, they'll be a person, a deeply, fundamentally, irreducibly human person, attempting the most extraordinary journey in the history of our species, carrying with them everything that makes us worth sending in the first place: our curiosity, our love, our ability to find meaning in suffering, our stubborn, irrational, magnificent refusal to stay where we were put. We have always been a species that walks toward the horizon. And the horizon, for the first time, is another world. Let me ask you something that sounds simple, but runs surprisingly deep. What time is it? Not on your phone, not on the wall, inside your body. What time does your biology think it is? Because here's the thing, your body keeps its own clock. Not one clock, actually. Trillions of them. Every single cell in your body contains a molecular timekeeping mechanism, a set of genes and proteins that cycle through a roughly 24-hour rhythm, orchestrating when to divide, when to repair, when to produce which hormones, when to be alert, when to rest. This internal timing system is called the circadian clock, from the Latin circa diem, meaning about a day, and it is one of the most ancient and fundamental features of life on Earth. Bacteria have circadian rhythms, plants have them, fungi have them, every animal that has ever been studied has them. This clock is so deeply embedded in biology that it predates multicellular life by hundreds of millions of years. It was conserved through every major evolutionary transition our lineage has undergone, through the first fish crawling onto land, through the age of reptiles, through the emergence of mammals, through 5 mass extinction events, through the entire adventure of primate evolution that eventually produced you. It is, in a very real sense, one of the oldest things about you, and on Mars, it is going to be in serious trouble. A Martian day, called a sol, lasts 24 hours, 39 minutes and 35 seconds. 37 minutes longer than an Earth day if you round it off. 37 minutes. That doesn't sound like much, does it? A little over half an hour. You might lose that much time in a long meeting. You might spend that long looking for your keys on a bad morning. But your biology doesn't experience 37 minutes the way your conscious mind does. Your biology experiences it as a daily miscalibration, an insult to an ancient system that has never, in its billions of years of existence, had to deal with a day that doesn't match the template. Think of it this way. Imagine your internal clock is set to ring every 24 hours on the dot. The alarm goes off, your body begins its morning cascade. Cortisol rises, body temperature climbs, alertness sharpens, digestive enzymes begin preparing for breakfast. All of this is choreographed. All of it is timed with extraordinary precision. Now imagine that the external cues your clock uses to stay calibrated, primarily sunlight, but also temperature cycles, meal timing, social interactions, now come 37 minutes late every single day. On day 1, the mismatch is trivial. You barely notice. On day 10, you've accumulated over 6 hours of drift. On day 100, you're nearly 62 hours out of alignment. On day 500, deep in the Mars surface stay, months away from any possibility of going home, the accumulated drift is enormous. And your body's internal clock may have fundamentally decoupled from the external environment in ways that cascade through every biological system you have. This is not a hypothetical concern. It is a documented, studied, serious challenge that mission planners and space medicine researchers have been wrestling with for years. NASA got its first sustained look at this problem, not on Mars, but on Mars missions by proxy. When the Pathfinder rover landed on Mars in 1997, and later during the Spirit and Opportunity rover missions in 2004, the ground control teams at NASA's Jet Propulsion Laboratory in Pasadena, California, made a striking decision. To operate the rovers most efficiently, the controllers needed to work on Martian time, scheduling their shifts so they were working during the Martian daytime at the rover's location. That meant their workday started 37 minutes later every Earth day. They wore special watches that ran on Mars time. They blacked out the windows of mission control to eliminate Earth's light-dark cycle. They shifted their meals, their sleep, their entire social lives to align with a Martian soul. Within weeks, many of them reported significant health effects: disrupted sleep, fatigue, cognitive fog, mood disturbances. Their families described them as disoriented, irritable, difficult to reach. Several controllers gained weight, as disrupted sleep is strongly associated with dysregulation of the hormones that control appetite, leptin and ghrelin. Some described the experience as feeling permanently jet-lagged, as if they were constantly flying across time zones without ever arriving anywhere. And these were people sitting in comfortable offices in Southern California, with access to their families, their doctors, their favorite restaurants, all the comforts of home. They could step outside and feel the California sun whenever they chose. The first human on Mars will have none of those anchors, and they won't be doing it for a few months of an exciting mission. They'll be doing it for 500 days. Let's make this concrete, because the cascade effects of circadian disruption touch virtually every system in the human body, and they are more serious than most people realize. Start with sleep. The most obvious casualty of a misaligned circadian clock is the quality of your sleep. Melatonin, the hormone that signals to your body that darkness has arrived and it's time to prepare for sleep, is produced by the pineal gland in direct response to the light-dark cycle perceived by specialized cells in your retinas. When that cycle shifts, melatonin production shifts with it. When it decouples entirely from a stable rhythm, melatonin secretion becomes erratic, sometimes rising at the wrong time, sometimes failing to rise adequately at all. Poor sleep, in turn, degrades almost everything else. The immune system weakens. Studies have shown that people sleeping less than 6 hours a night are 4 times more likely to develop a cold when exposed to a virus than those sleeping 7 hours or more. Cardiovascular stress increases. Chronic sleep deprivation is associated with elevated blood pressure and inflammation markers. Metabolic function suffers. Insulin sensitivity drops, increasing the risk of blood sugar dysregulation. Pain perception increases. The same injury or discomfort registers as more severe in a sleep-deprived brain. And crucially, for an astronaut who needs to be sharp every single day, cognitive performance degrades profoundly. reaction time slows, working memory falters, decision-making quality declines, emotional regulation, the ability to stay calm under pressure, to respond thoughtfully rather than reactively, erodes. Behavioral research from NASA indicates that the majority of operational errors documented during astronaut analog studies could be attributed to sleep-related impairment. Now, move beyond sleep. Circadian rhythms regulate the timing of virtually every hormonal cycle in your body. Cortisol, the stress hormone that, in proper rhythm, peaks in the morning to help you mobilize energy and falls through the day to allow recovery, loses its clean daily pattern under circadian disruption. The result is a state of chronic low-grade physiological stress. Even in the absence of any specific stressor, your body is running an alarm system that has lost its off switch. Testosterone, growth hormone, thyroid hormone, insulin, all of these follow circadian patterns that can be thrown into disarray by a misaligned clock. The gut microbiome, which has its own circadian behavior, shifts. The immune system's daily patrol rhythm, when certain immune cells are most active and where they're deployed in the body, gets scrambled. Even DNA repair follows a circadian pattern. The molecular machinery that fixes radiation damage to your genome is more active at certain times of day and less active at others. If you're receiving radiation, as a Mars crew certainly will be, and your repair machinery is running on a garbled schedule, the compounding of radiation damage and impaired repair creates a risk that is greater than either factor alone. Here's something that I find extraordinary and that most discussions of Mars missions completely overlook. The Sun, as seen from Mars, is not the Sun you know. It is smaller, noticeably smaller. From Mars, the Sun subtends an angle of only about 2/3 of a degree across the sky, compared to 1/2 a degree as seen from Earth. That sounds like a small difference, but visually it's enough to be disorienting. The Sun looks shrunken, diminished, less authoritative, and its light, already filtered and scattered by the thin Martian atmosphere and ever-present dust, is dimmer and redder. Mars receives only about 43% as much solar energy per square meter as Earth does. The spectrum of light reaching the Martian surface is different, more red, less blue and violet. And this matters enormously for the circadian system because the specialized photoreceptive cells in your retinas, called intrinsically photosensitive retinal ganglion cells, are most sensitive to short-wavelength blue light, roughly in the 480 nanometer range. For the purpose of circadian calibration, the Martian surface, with its reddish dust and dimmer, warmer spectrum sunlight, will provide less of this blue light signal than your circadian system is adapted to receive. The result is a weaker, less reliable environmental time signal. The clock gets harder to set, the drift accelerates. Researchers are well aware of this. One proposed countermeasure involves specially designed lighting systems inside the Mars habitat, lights capable of delivering the specific wavelengths and intensities needed to keep the crew's circadian systems properly entrained, essentially simulating the light environment the human brain was designed for. Timed carefully, these systems could function as artificial suns, keeping the crew's biology anchored even when the real sun outside can't do the job adequately. But this solution depends on equipment working flawlessly for years. On a planet where dust storms can last for months, where everything mechanical is under extraordinary stress, and where there are no spare parts stores around the corner. Now let's zoom out for a moment, because I want to place all of this, the circadian disruption, the radiation, the isolation, into the bigger picture that I think is the most important frame for understanding what the first human on Mars will actually go through. Evolution is not a plan, it is a response. It is nature's way of solving problems after the fact, identifying organisms that survived a particular challenge and passing on whatever made them survivors to the next generation. For 300,000 years, Homo sapiens evolved on Earth. The 24-hour day shaped our biology as surely as oxygen shaped our metabolism. The gravitational pull of one Earth gravity shaped our bones and cardiovascular system. The electromagnetic spectrum of our yellow dwarf star shaped our vision and our sleep. The microbial community of our ancestral environment shaped our immune system. The social structures of small tribal groups shaped our nervous systems and our deepest psychological needs. None of these conditions exist on Mars. And yet, and this is the thought I want you to sit with, every single challenge I've described has a parallel in Earth's evolutionary history. Ancestral humans survived ice ages that drove dramatic changes in available light and temperature. They crossed mountain ranges that tested cardiovascular adaptation. They colonized every climate from equatorial rainforests to Arctic tundra, adapting their biology, their culture, and their technology as they went. The challenges of Mars are more extreme than any of these. But they are not different in kind, they are different in degree. They are the same fundamental conversation between biology and environment, turned up to a volume no member of our species has ever had to endure before. And here is what I believe, based on everything the science is telling us. The first human on Mars will not be broken by this conversation. They will be transformed by it, not all at once, not without suffering, not without cost, but transformed nonetheless in their biology, in their psychology, in their sense of what they are and what the universe is and what their place within it might be. Think about what it would feel like to watch a sunset on Mars. The sky turns blue around the setting sun, the reverse of what happens on Earth, where sunsets go red and orange. This is because of the way Martian dust particles scatter light, large, iron-rich particles that scatter shorter wavelengths around the sun more effectively than longer ones, producing a pale blue halo in exactly the spot where earthly skies would glow amber and rose. The rest of the sky, away from the sun, is the characteristic butterscotch of a typical Martian afternoon. It's alien, deeply, disquietingly alien, and yet it's a sunset, a star going down behind a curved horizon, a planet turning away from its local light source. The same fundamental phenomenon you've watched from a beach or a mountain, the same geometry that poets have written about for thousands of years. Different in every detail, identical in its cosmic architecture, there is something in that image that captures, for me, the entire story of what the first human on Mars will experience. Everything will be different. The sky, the gravity, the silence, the cold, the radiation, the light, and underneath all of it, the same ancient human standing on a planet watching a star set, wondering what comes next. Your circadian clock will struggle, your immune system will adapt, your bones will work harder than they ever have, your mind will be tested in ways no psychologist has fully mapped. Your sense of time and self and home will shift, but you will still be there, watching the sunset, carrying within you 3 billion years of life's accumulated wisdom about how to survive in a difficult universe. That wisdom is not nothing. In fact, it might be everything. You are not alone. You have never been alone. Not for a single second of your life. Right now, as you watch this, as your eyes move across the screen, as your heart beats and your lungs expand, you are carrying an entire civilization inside you, trillions of living organisms representing thousands of distinct species, occupying every surface of your body that touches the outside world, your skin, your mouth, your lungs, your reproductive tract, and most densely, most consequentially, your gut. This is your microbiome. And it is not a passenger, it is not a hitchhiker that happened to climb aboard and never got off. It is a partner, ancient, deeply integrated, essential, that your biology has co-evolved with for so long that the boundary between where you end and where it begins is, in the most literal biological sense, genuinely blurry. Consider this number. The human genome contains approximately 20,000 protein-coding genes. The collective genome of your gut microbiome, scientists call it the microbiome, contains an estimated 2 to 20 million genes. Depending on the individual, your microbial partners are doing biochemistry your own cells simply cannot do. They break down dietary fibers your digestive enzymes can't touch, producing short-chain fatty acids that feed the cells lining your gut wall and regulate inflammation throughout your body. They synthesize vitamins, particularly vitamin K, and several B vitamins that you cannot make yourself. They metabolize bile acids in ways that affect cholesterol levels, hormone balance, and fat absorption. They are, in a very real sense, organs, distributed microbial organs that you cannot see, cannot feel directly, and for most of human history had no idea existed. And when you board a rocket to Mars, every single one of them comes with you. The question of what happens to the human microbiome in deep space is one of the most active and genuinely urgent areas of space medicine research right now. Because unlike bone density, which we have countermeasures for, and unlike radiation, which we can partially shield against, the microbiome is a vastly complex living system that we are only beginning to understand. Manipulating it is still more art than science, and the consequences of getting it wrong are serious in ways that connect to almost every other biological risk we've talked about. Here's what we already know from International Space Station research. Microbial diversity in the gut decreases during long-duration spaceflight. Diversity is the gold standard of a healthy microbiome. The more different species you have operating in balance, the more resilient and functional the system tends to be. A diverse microbiome is better at recovering from disruptions. A less diverse one is more vulnerable to being thrown out of balance by pathogens, by stress, by dietary changes. The specific bacterial populations that shift are telling. Bacteria from the genus Firmicutes, which includes species associated with efficient energy extraction from food, and which when overly dominant have been linked to obesity and metabolic syndrome on Earth, tend to increase in proportion during spaceflight. Bacteria from Bacteroidetes, associated with leaner metabolic profiles and certain anti-inflammatory functions, tend to decrease. This is not a trivial shift. The ratio of Firmicutes to Bacteroidetes is one of the most studied parameters in gut microbiome research because of its associations with metabolic health, inflammation, and immune function. At the same time, potentially pathogenic microorganisms, bacteria that under normal circumstances live in small numbers and cause no problems, kept in check by the competitive balance of the broader microbial community, can expand their populations when that balance is disrupted. Studies on Salmonella, grown in simulated microgravity at NASA's Johnson Space Center, showed that the bacteria became significantly more virulent, better at infecting cells, more resistant to immune responses, requiring a lower dose to cause infection. The mechanism appears to involve changes in gene expression driven by the fluid dynamics of microgravity, which alters the mechanical signals bacteria use to sense their environment and regulate their behavior. Think about what this means. You board the spacecraft with a perfectly healthy gut. The journey takes 9 months. Over those 9 months, with no fresh food, no natural environmental microbial exposure, recycled air, disrupted sleep, radiation stress, and the physical effects of microgravity, your internal ecosystem begins to drift. The diversity drops, the balance shifts. Opportunistic organisms grow bolder. By the time you reach Mars, the civilization inside you is not quite the one that left Earth. Now let's connect this to something you might not expect. Your mind. The gut-brain axis is one of the most significant scientific discoveries of the past 2 decades. It refers to the bidirectional communication highway between your gut microbiome and your central nervous system, a network of biochemical signals, neural pathways, and immune messages that allows the state of your gut to directly influence the state of your brain, and vice versa. The vagus nerve, the longest cranial nerve in your body, running from your brainstem all the way down through your chest and into your abdomen, is the primary physical conduit of this communication. Approximately 80% of the signals traveling along the vagus nerve go from the gut to the brain, not the other way around. Your gut is, in a very real neurological sense, talking to your brain far more than your brain is talking to your gut. What is it saying? Your gut microbiome produces and influences the production of an extraordinary range of neuroactive compounds. Serotonin, the neurotransmitter most associated with mood regulation, feelings of well-being, and emotional stability, is produced in the gut at a rate of roughly 90% of your body's total supply. The microbiome influences serotonin production through its effects on the enterochromaffin cells lining the gut wall. When the microbiome shifts, serotonin production can shift with it. Gamma-aminobutyric acid, or GABA, the brain's primary inhibitory neurotransmitter responsible for calming neural activity, reducing anxiety, and promoting sleep, is produced by certain gut bacteria. Specific strains of Lactobacillus and Bifidobacterium have been shown to produce GABA directly and to stimulate its production in gut cells. Studies in germ-free mice, animals raised without any gut microbiome at all, showed dramatically elevated stress responses, anxiety-like behaviors, and disrupted social interactions compared to normal mice. When those mice are colonized with specific bacterial strains, some of these behavioral changes reverse. Short-chain fatty acids produced by microbial fermentation of dietary fiber, particularly butyrate, cross the blood-brain barrier and influence the function of microglia, the brain's resident immune cells. Microglia sculpt neural circuits, regulate synaptic pruning, and respond to injury and infection in the brain. Their function is directly affected by the metabolic output of your gut bacteria. What this means for a Mars astronaut is this: the psychological resilience of the first human on Mars will not be determined solely by their mental training, their personality profile, or their support network. It will be determined in part by the health of the ecosystem in their gut. A disrupted microbiome doesn't just increase the risk of infection or digestive problems, it can directly alter mood, anxiety levels, stress responses, and cognitive function, the very capacities that a Mars crew will depend on most desperately. This is not fringe science. This is published, peer-reviewed, increasingly well-established biology. And it is one of the most compelling reasons why the nutrition and microbiome management protocols for a Mars mission may matter as much as the engineering of the habitat itself. Food on Mars is not a solved problem. This might seem like a strange place to arrive in a conversation about the microbiome, but it is absolutely central, because the single most powerful influence on the composition of your gut microbiome, more than stress, more than antibiotics, more than almost anything else, is what you eat. Dietary fiber is the primary fuel source for the beneficial bacteria in your gut. Specifically, prebiotic fibers, complex carbohydrates that human digestive enzymes cannot break down, which pass intact to the large intestine where gut bacteria ferment them, are what keep the beneficial populations fed, diverse and dominant. Fruits, vegetables, legumes, whole grains, nuts. These are the building blocks of a healthy microbiome. On a Mars mission, at least in the near-term scenarios being planned, the crew will rely primarily on pre-packaged, processed foods with long shelf lives. The NASA Space Food System has made enormous advances in nutrition and palatability, but there are hard limits on how much fresh produce can be carried, and pre-packaged foods are generally lower in the diverse prebiotic fibers that a thriving microbiome requires. This is not hypothetical damage. Research on the diets of International Space Station crew members has already documented microbiome changes, consistent with reduced dietary diversity. The longer the mission, the more pronounced these changes become, which is why one of the most exciting areas of current Mars mission preparation involves growing food on Mars itself. NASA's Veggie project, which has been running experiments on the International Space Station since 2014, has successfully grown lettuce, radishes, mustard greens, kale, mizuna, and dwarf wheat in space. Astronauts have eaten crops grown in orbit. In 2020, the crew of Expedition 64 harvested radishes grown aboard the station, the most complex crop yet attempted in microgravity. The Mars surface, while obviously not farmable outdoors, contains the raw materials for growing food in pressurized greenhouses. The soil, called regolith, is rich in minerals, though it also contains perchlorates, a class of oxidizing chemicals toxic to humans and plants, that would need to be removed or neutralized before the regolith could support plant growth. Researchers at various institutions have been working on exactly this problem, testing methods of perchlorate reduction using specific soil bacteria that metabolize perchlorates as an energy source. Sunlight on Mars, while dimmer than on Earth, is sufficient for plant growth with the right crop selection and supplemental artificial lighting. Water ice, detected in enormous quantities beneath the Martian surface and at the poles by instruments including the MARSIS radar aboard the Mars Express orbiter and the SHARAD instrument on the Mars Reconnaissance orbiter, could in principle be extracted and used for irrigation. A functioning greenhouse on Mars would do something extraordinary. It would produce fresh food, fiber-rich, microbiome-supporting, nutritionally dense fresh food on another planet for the first time in human history. It would recycle carbon dioxide into oxygen through photosynthesis, reducing the burden on mechanical life support systems. It would provide psychological benefits, the sight of green, growing things in an otherwise utterly barren landscape is not trivial for human well-being. Research on Antarctic station crews has shown that tending plants reduces stress markers and improves reported mood, and it would begin in a small but meaningful way the transformation of Mars from a place humans visit into a place humans inhabit. Let's take a step back from the cellular level and look at the whole organism. You, the first human on Mars, you've arrived. The rocket has landed, the dust has settled, the habitat is pressurized and functional. You have survived 9 months of deep space travel, absorbed your dose of radiation, adapted your cardiovascular system, and managed the first stages of circadian disruption. You have landed, now you have to live there. And one of the first things you will notice, one of the things that no amount of simulation on Earth can fully prepare you for, is the silence, not just the quiet of an isolated environment, the silence of a world with no biosphere. On Earth, even in the most remote wilderness, there is biological noise, wind moving through leaves, the call of a bird, the buzzing of an insect, the rustle of something moving in the undergrowth. Life is everywhere and life is loud, and we are so immersed in that noise that we don't register it as noise at all. It is the background hum of our home. Mars has none of this. The wind moves sometimes violently, in global dust storms that can last for months, but it moves through a landscape of rock and dust and ancient craters with no living thing to interact with. The silence is not peaceful in the way a quiet room is peaceful. It is absolute. It is the silence of deep geological time. It is the silence of a world that has been waiting, empty, for 4.5 billion years. How will a human nervous system, evolved over 300,000 years in the noisiest biosphere in the known universe, respond to that silence? We don't know. No one has ever heard it. No recording from our rovers comes close to conveying what it would actually sound like or feel like to stand in it. But here's a thought that strikes me as profoundly important. The first human to hear that silence will carry within them the sound of every environment their ancestors ever inhabited. The savannas of East Africa, the forests of Europe, the Arctic tundra, the river valleys where the first cities rose, all of that biological history encoded in a nervous system that has never before encountered a world without life. And in that moment of silence, standing on the rust-red surface of another planet, they might hear something that no human has ever heard before, the sound of being the only living thing, as far as any instrument can detect, on an entire world. That is a moment I think about often, not with dread, with awe, because what comes next, what that astronaut does in the moment after that silence lands on them, might be the most important behavioral data point in the history of human psychology. Do they feel terror, loneliness, a bone-deep homesickness for a planet 200 million miles away? Do they feel something unexpected, wonder so vast it silences the part of the brain that generates fear? Do they fall to their knees or stand straighter than they ever have before? We don't know, nobody knows, because nobody has ever been there. But here's what I believe, based on everything the science tells us about human adaptability, about the microbiome's role in mood and resilience, about the circadian system's capacity to recalibrate under the right conditions, about the psychological transformations documented in people who have pushed themselves to the absolute edge of human experience. The first human on Mars will find a way to be okay, not easily, not all at once, not without grief and strain and biological cost, but they will find it. Because finding a way to be okay in impossible circumstances is quite literally what our species does. It is what we have always done. Our microbiome adapted to every new continent our ancestors colonized, our circadian clocks adjusted to every new latitude, our immune systems learned the local pathogens wherever we settled. Our minds built meaning out of every new landscape, however alien, however harsh. Mars is more alien and more harsh than any of those previous challenges by orders of magnitude. But the biological toolkit our species carries is the same toolkit that handled all of them. It has been tested for hundreds of millions of years. It has survived every challenge the Earth could throw at it. Now it gets to find out what else it can do. And so, here we are, at the edge of the most audacious chapter in the entire human story. Everything we've explored together, the DNA negotiating with cosmic radiation, the microbiome learning a new world, the circadian clock searching for a rhythm in an alien sky, the mind bending under the weight of absolute isolation and absolute silence. None of it is a story of fragility. All of it, every single biological conversation, every cellular adaptation, every psychological transformation, is a story of a species that was never content to stay where it was put. The first human on Mars will carry 3 billion years of life's hard-won wisdom in every cell of their body, and they will carry the hopes, the curiosity, and the love of every human being who ever looked up at the night sky and wondered. They will be cold and tired and far from home in a way no words can fully capture, and they will be standing on another world. If that doesn't fill you with the kind of awe that makes the ordinary world feel suddenly, overwhelmingly precious, then nothing will. Thank you for going on this journey with me. If you made it this far, you are exactly the kind of curious, open-minded person this channel exists for. So please subscribe, leave a thumbs up, and tell me in the comments, if you could be the first human on Mars, would you go? See you in the next one.